So our next speaker is Professor Jo Cambridge from UCL, but I'm pleased to say she's actually an Australian. And she's from Perth originally, apparently. She's a B-cell immunologist, and her focus is on autoimmune diseases, which obviously can overlap into ME/CFS.
Thanks, Paul. Thank you very much. Thank you for having me here and presenting this work. Now I'm going to present all the work I'm going to present is by Fane Mensah, my PhD student. I only came into this field about 4 or 5 years ago, and the thing that struck me after I reviewed the literature, the immunological literature, was that there was very little consistency, shall we say, throughout. And the most consistent finding was changes in energy metabolism. So Fane's thesis was based on the premise that we could measure changes in metabolic thresholds in patients with ME/CFS by following B-cell responses to stresses in vitro. Now, peripheral blood B cells are an extremely dynamic lymphocyte population. Every day, 10 to the 9 new B cells come out of the bone marrow. They start circulating around, and then if they're not given the right signals to survive, they die. Once they're exposed to antigen and other sort of co-signals, they can develop into sort of mature naive cells or memory cells, and ultimately many of them will progress to be antibody-producing cells. And you can measure these by using a combination of markers shown here, IgD and CD27. You can measure these relative expression by FACS analysis, and using another marker, CD38, you can look at additional, another 2 additional populations, these very early ones here that come straight out of the bone marrow, and the plasmablasts or plasma cells. Okay, so the first thing we did, that Fane did, was to look again at the classical B-cell phenotypes, immunophenotypes in patients. And this just shows a representative plot of the naive and memory populations. You can distinguish by looking at the relative expressions of IgD and CD27. Here are the naive ones, pre-switch memory, and with D38, the additional ones here, the transitional and the plasmablasts. So he looked at 32 patients with ME/CFS versus 38 patients with ME/CFS and 32 patients with age-matched healthy controls. It's very important in any studies of B cells that you age-match very precisely, because the markers change with age. And so we found absolutely no difference in all these 10 different subpopulations between the patients and the controls. But then we looked at an extended immunophenotype, looking within each individual sort of quadrant, if you like, looking at additional markers that we thought might be relevant. And here we did find some differences. We found that this molecule called CD24 was significantly increased, both in terms of the percentage of cells expressing it and in the amount of receptors per cell in the ME/CFS patients. We thought this was quite interesting, but we didn't know what it meant. And we found that the populations expressing the highest amounts of this CD24 were in these naive and pre-switched populations. And just showing the data here, you can see the ME/CFS patients are in purple, and you can see that the values are higher in these populations here, in the transitional, naive, and the pre-switch memory population, compared to the healthy controls. And we've repeated this in several different cohorts now. So we found this increased percentage of these CD24 B cells, particularly of the memory type, and we also found another cell type that was increased as well, but that's all published, and I haven't got time to go into that. Okay, so in conclusion, our immunophenotype analysis showed that the CD24 is overexpressed, and this CD24 positive population also has a marginal zone type phenotype, which is very important in early responses to viruses, and also as a sort of link between the innate immune response. Okay, now CD24 is an extremely interesting molecule. It is a GPI-linked glycoprotein, which means it can then move in lipid rafts in different ways with other receptors. So it can co-localize with a whole number of different receptors. So it's got many different co-signaling potentials. It's associated expression-wise with immature cells, particularly B cells. It plays a very important role in early B cell development, where cross-linking it will actually cause apoptosis, but that's in the pro-B cell stage. It's also a sort of guiding molecule on differentiating neuroblasts in the brain. Again, has a quite complicated relationship with neural development. The oncology people are very interested in CD24 because very high expression is related to increased in cell adhesion and invasion of tissues. And it's also linked with resistance to getting rid of drugs, you know, through the ABC transporter. Now, its functions in B cells, as I said, it's very important in the early B cell development for controlling growth. And in the brain and gut, in fact, it's important co-stimulator for T cells. In those environments are low in CD28 positive T cells, and so CD24 can sort of like be a co-stimulator in those particular situations. Okay, so what about the number of receptors of CD24 on these B cells? What happens during development in this dynamic development stage? Well, after the early stage, CD24 goes down in the naive cells, and then we found it actually rises again on the memory cells. So there's a lot of work looking at the CD24 in the naive cells, but very little on the memory cells. So we were interested in what was going on. So we looked to see what happened following in vitro stimulation to CD24. So there's many ways of stimulating cells, B cells in culture, and we use the usual, the ones T-independent stimulation using CPG, where you could measure, this is a very strong proliferation signal, and you get IgM produced, and T-dependent stimulation with CD40, proliferation, you get differentiation, also IgG production. Okay, so we looked at CD24 expression after 6 days culture, and again, we found that the B cells from the ME/CFS patients here still continued to express more CD24 in the unstimulated, but not the stimulated culture. So this is 6 days of culture. So these unstimulated cells retain their basal higher expression of CD24. And then we looked at a time course, see what happened in between days 0 and 6. And if you just look, it's easy to see on the right-hand one, this is T-dependent stimulation. Again, the ME/CFS patients are in red, and you can see days 0, 3, and 1 and 3, very significantly higher retention of this CD24 on the B cells from the ME/CFS patients compared to controls. And the same thing happened with the T-independent. So there's this significantly delayed loss of CD24 from the B cells compared with the healthy controls. So then we thought, well, why could this be significant? So we know that CD24 is very important in oncology, in prostate cancer, for example. It's been shown to be linked with metabolism, where inactivation of AMPK is associated with decreased CD24 expression, and also late senescent B cells, DNEG 27 negative, is associated with spontaneous phosphorylation of AMPK, and we've heard about AMPK earlier today, an alternative energy supply. So what about in these ME/CFS B cells? Is there any association with AMPK? So we know that low ATP levels and stress activate phosphorylation of AMPK. So there's a whole load of different stimuli will do this. And therefore, phosphorylation of AMPK controls signaling pathways that will help replenish ATP supplies very basically. So we looked then at the relationship between naive B cells, memory B cells, and CD24 expression with phosphorylation of AMPK. Again, we did this with phosphoflow. If you look at this axis here, this is phosphorylation of AMPK in naive B cells here. This is AMPK in memory B cells. These quadrants here represent 24 positive. And you can see in the naive cells, there's no phosphorylation of AMPK here. But with the memory B cells, the 24 positive quadrant, you can see that AMPK is phosphorylated. So the CD24 memory cells utilize AMPK as an alternative energy source. So after culture, as I've described, there's a significantly delayed loss from the B cells, and increased CD24 expression is associated with metabolism. But how is CD24 recycled? Now, this is a very interesting pathway. It's a very unusual way of recycling a molecule. Usually things are either cut off or degraded or taken in or recycled. But CD24 recycling is by formation of microvesicles budding off from the cell surface, constitutively usually, but increased after stimulation. And these little microvesicles are very selective in the protein cargo, the protein RNA and surface receptor cargo they carry. They're very selective. I haven't got time to go into those, but they're particularly enriched in certain mitochondrial and metabolic proteins, particularly after B-cell receptor or CD24 stimulation. And in fact, it's been suggested that this CD24 packaging plays quite an important role in regulating mitochondrial health after proapoptotic stress. Okay, so the next, that's the first stage. I'm just finishing with CD24 there. The thing about ME/CFS, as you've seen, is it's really when you perturb the system that you can more easily measure the problems underlying the condition. So Fane and Chris Armstrong from the University of Melbourne worked together to set up an in vitro system to study energy metabolism in relation to B cells, using B cells stimulated in the ways I just talked about. The investigations included these here. I'm just going to go through them very quickly one by one. The first thing, we looked at mitochondrial mass. Now, Paul mentioned mitochondrial mass earlier, saying, finding that it wasn't different. So what we've done here is we used MitoTracker. Again, we use phosphoflow for this. This is MitoTracker red staining mitochondria here. This is by FACS. This is relative expression here. And we find that mitochondrial mass increases, as you'd expect, when you go from naive to memory B cells. Okay. And then looking at the patients with ME/CFS compared to healthy controls over cycles of proliferation. Now, this is, if you just look at this one here, T-dependent stimulation, which is higher, you get higher mitochondrial mass than T-independent. And you can see consistently across, this is 4 cycles of proliferation. You can see significantly lower mitochondrial mass in the ME/CFS patients compared to the healthy controls. And that was the same with T-independent stimulation as well. So this was highly significant and reproducible. The next phase, the next test that they set up was a very innovative way of looking at cells over a period of culture, sampling cells in this upper chamber here. This is a sort of a pervious chamber here where you put the cells in the top into the medium here. You can sample the medium, you can sample the cells at various days of incubation and measure the markers on the B cells by flow cytometry, for example. And Chris measured the metabolites using NMR. He'll be telling you more about that later. and just show the sorts of things you can do. Well, firstly, this is just an example of cell growth. Oh, I don't know if you can see this very well. This is an example of cell growth. This is forward scattered, so this is the size of the cells, and this is how granular they are. And if you just look at the yellow going to red, well, you can't really see this. It's just showing that the most active phase of cell growth is around here about day 3. And that's all we can see on that one. Can't see that very well. But of course, when you're looking at metabolites, you must take into account the fact that there's cell death going on. There are very bits of debris around that will all contribute to the sort of milieu of what you're measuring. So you have to be very precise and careful about what you're relating your findings to. And so Chris and Fane have established an excellent system for looking at the fold change in live cells, dead cells and debris, comparing healthy controls with ME/CFS patients. This looks significant, but it isn't. But there is a sort of trend towards, between days 1 and 3, more dead cells in the ME/CFS patients. 2 minutes? Just about done. And then you could also look at the immunophenotypes on the cells at the same time as the metabolites. For example, this is CD24, and you can see fold change in glucose here, which correlate, but there's no correlation of fold change glucose with any of the other receptors. Okay. And so just the final thing, the other thing you can look at is the change in amino acids in the culture supernatants. This here is fold change, and here you can see there's differences between the healthy controls and the ME/CFS patients. Okay, so in conclusion, the higher expression of CD24 on these patients shows that there's an increased need to switch to alternative sources of ATP generation, perhaps through AMPK. And retention may be indicative of defective regulation of mitochondrial health. The decreased mitochondrial mass in the patient's B cells over cycles of proliferation, again, confirm this inability to meet the energy demands. And the amino acid usage is also of interest, and it seems to be very selective. So finally, can we use this for biomarker purposes? Maybe. I think we possibly can. And I think it's also of use to think of it as a good way of investigating underlying cellular metabolic pathways that may be defective. So I want to thank Emerge very much for getting me back home, and for the funding support from Invest in Me and Solve ME initiative. As I say, all the work was done by Fane, my wonderful PhD student, and Chris in Australia. Thank you very much.
We've got time for a few questions. So, Brett, I think, had his hand up first.
Yeah, thank you, Heidi. Thank you very much. I have, a question in 2 parts. On a technical sense, you showed the data with the mitochondrial mass. That was a 6-day culture period.
Yeah.
So some of the cells died, some stayed alive. How did the ones that stay alive stay alive? And what culture medium do you use in these assays? This is something I've discussed with Paul as well. And my second part of the question is, I've always wanted to ask a real immunologist this question. Do you think ME/CFS is an autoimmune disease?
That's the second one, no. Most pays. The first question is, with the mitochondrial mass experiments were done just usually, we do live dead cell staining as well, so that we're just looking at the live cells there. The culture medium, just the standard RPMI 1640 fetal calf serum.
10% fetal calf.
10%. And we measured the cycles of proliferation using CFSE. So then we, looked at the mitochondrial mass at all the different, I can show you the figure later looking at that. So does that answer your question? I'm a bit jet lagged.
No, pure B cells.
Oh yes, absolutely. We use pure B cell cultures. Yes.
Both you and Paul referred to the production of immune cells in the bone marrow. You even quoted a 10 to the 9th cells per day coming in. And one of the questions that we always talk about is, how is a phenotype maintained in a cell population that is turning over much faster than the time course of the disease?
How is the phenotype? Well, the phenotypes are not, it changes continuously, but at different stages of development, because you've got regulated expression of genes at the different stages. We just call them different things.
Yeah, I just want to make it clear whether the differences you see in CFS can be maintained without the cause being in the bone marrow.
Oh, I see what you mean. Probably not. No. So it's very interesting because, I mean, I work on rheumatoid arthritis, and I would say, if it was rheumatoid, I'd say no. But with ME/CFS, I don't have enough information to answer that question. But I do think there may be soluble factors in the mix, because if you looked at those graphs, you can see after 3 days there seems to be like a washout period, whereas the cell is now able to restore homeostasis, if you like, to the CD24 expression. So that's a very interesting question, and I mean, I'm very much for bone marrow driving most diseases.
Okay, well, we won't keep you from your lunch anymore.
No.
So thanks to all the speakers for great presentations. I think it's been really enlightening.